A colony in microbiology is an aggregate of cells arising from a single parent cell. This is useful to us because they are clones derived from that parent cell. It can be difficult to test or identify a single cell, but using a colony will often give us enough cells to work with in the identification process. All cells have requirements for growth. Nutrients will provide carbon, oxygen, hydrogen, and nitrogen the cell needs. All cells must have a source of carbon, a source of energy, and a source of electrons. There are different ways different organisms go about obtaining these things. An autotroph uses inorganic sources for carbon, such as carbon dioxide. Chemotrophs will use redox reactions of either organic or inorganic compounds as an energy source, where a phototroph will use light as an energy source. When more than one of these categories applies, the terms are combined. For example, a photoautotroph will use light for energy, but carbon dioxide for carbon. The chemoautotroph uses carbon dioxide for carbon and catabolizes organic molecules for energy. The photoheterotroph uses light for energy and catabolizes organic compounds for nutrients. A chemoheterotroph uses organic compounds for both energy and carbon. The organotroph requires electrons from the same organic molecule that provides carbon and energy. The lithotroph acquires electrons from an inorganic source such as hydrogen sulfide. Organisms differ on how they deal with oxygen. For the obligate aerobe, oxygen is essential and acts as a final electron acceptor. At the other end of the spectrum, oxygen is deadly for the obligate anaerobe. There are several toxic forms of oxygen, which aerobes have means to deal with and anaerobes do not. The singlet oxygen, or molecular oxygen with an electron boosted to a high energy level, is a very reactive oxidizer. The carotenoids will remove the excess energy. An example would be the antioxidant vitamin A, which is a source of beta-carotene, a carotenoid. The superoxide radical undergoes incomplete reduction of the oxygen during the electron transport chain. Aerobes produce superoxide dismutase to detoxify them. Another toxic form is the peroxide anion. The enzyme catalase converts 2 hydrogen peroxides to 2 waters in molecular oxygen. The use of this reaction can be used in the catalase test to help identify organisms. Peroxidase converts hydrogen peroxide with NADH plus hydrogen ion into 2 waters and NAD+. Ionizing radiation can form the very reactive hydroxyl radical as another toxic form of oxygen. The facultative anaerobes have the option of doing fermentation or anaerobic metabolism if needed, but they are less efficient without oxygen. The aerotolerant anaerobes do not do aerobic metabolism, but tolerate oxygen because they have enzymes to handle the toxic forms of oxygen. The microphiles require oxygen in minute amounts of only 2 to 10% and are damaged by the 21% oxygen present in the atmosphere. Nitrogen is another important element. During nitrogen fixation, nitrogen gas from the air is converted to ammonia to provide a usable form of nitrogen to other organisms. Carbon, hydrogen, oxygen, and nitrogen make up 95% of the cell, but cells also use phosphorus, sulfur, calcium, manganese, magnesium, copper, iron, and other trace elements in very small amounts. Temperature is important because it will influence the 3D structure of proteins and lipid components of the cell. Microbes are found in virtually all places and temperatures on Earth. The minimum temperature for an organism is the lowest temperature metabolism is possible, but they may survive lower temperatures. This is why freezing food will allow you to store it longer by inhibiting microbe growth, but it does not necessarily kill the organism. The maximum temperature is the highest temperature where metabolism is possible. Higher temperatures may permanently denature structures, damaging the cell. The optimum temperature is where the metabolism is highest. For many pathogenic bacteria, the optimum temperature is 37 degrees Celsius, or human body temperature. The psychrophiles grow best at temperatures below 15 degrees Celsius. These organisms will grow in snow, ice, and cold water. Psychrophiles can cause the snow to appear pink on glaciers. The mesophiles grow at 20 to 40 degrees Celsius. Most human pathogens are mesophiles because of the body temperature being 37 degrees Celsius. The thermophiles will grow in temperatures above 45 degrees Celsius. These organisms will grow in places like composts or hot springs. Hyperthermophiles thrive at temperatures above 80 degrees Celsius. These tend to be in submarine hot springs where the pressure allows the water to get above boiling temperature at the sea level of 100 degrees Celsius. The pyrodictium grows naturally at 113 degrees Celsius and will survive up to an hour at an autoclave at 121 degrees Celsius. Generally, things are not even autoclaved for an hour. There is a link here that you can follow that's also on Angel to read more information on some hyperthermophiles. The pH can affect the type of microbe growth because it can interfere with chemical bonding. Neurophiles grow in a pH range of 6.5 to 7.5, which is also the pH range of most tissues in the body. The acidophiles like an acidic habitat, like the Helicobacter pylori found in the stomach. Alkalinophiles thrive in alkaline habitats. The Vibrio cholera, causing cholera, grows best at a pH of 9. Water can also affect microbial growth. Osmotic pressure is the pressure exerted on a semi-permeable membrane. When the cytoplasm shrivels due to water loss, we have plasmolysis. The concentration of salt in the environment can alter osmotic pressure. The obligate halophiles are adapted to growth under high osmotic pressure in places such as the Great Salt Lake. Because most organisms cannot handle the osmotic pressure, food dehydration and salting food becomes a viable means of preserving certain foods. Some organisms can tolerate 30% salt. Staph tolerates up to 20% salt, so it is able to grow on skin, even after profuse sweating. The barophiles live under extreme pressure. These are found in the deep ocean. For every 10 meters of water depth, the pressure increases by 1 atmosphere. Ecology looks at how different organisms live in association with other organisms. Microbes may form biofilms. In a biofilm, we have a different species that will attach, or different groups of species that will attach as a group and display different metabolic and structural traits than they would if they were on their own. Here's a video you can watch on your own that is a time-lapse of biofilms growing on human teeth, as well as 2 other videos here that you can either watch through the lecture notes or through the links on Angel. Bacteria can be quorum sensing, where they will respond to the density of nearby bacteria. The ecology can influence the ability of an organism to cause disease and suggests our friendly flora or good bacteria living in our body can be extremely important in preventing and fighting disease. There is also a link here where you can watch a video clip on the extremophiles.